Today I'd like to go over the Unit 3 study guide on energy. So let's just jump right into this. Number one, organic compounds, organic molecules, and biomolecules all are the same thing, and they all contain the element carbon. There are 4 types of organic molecules, and there's actually a mistake. There's only 3 boxes. They are carbohydrates, lipids, I'm going to make another box, proteins, and nucleic acids. Number 3, the monomer, which is the building block or subunit of a carbohydrate, is a monosaccharide. Number 4, another word for carbohydrates is, and it starts with an S, is sugar. Sugars are carbohydrates. Number 5, a bunch of sugars linked together is called a blank. And it's, again, a hint, it starts with a P. It is a polysaccharide, which is a large polymer. Number 6, this example of a carbohydrate is a monosaccharide and looks like a ring with 6 sides. Hint, it starts with a G. That would be glucose. Number 7, this example of a carbohydrate is a polysaccharide. It is found in cell walls of plants and is used for structural support, that is cellulose. Number 8, this example of a carbohydrate is a polysaccharide and is used to store extra energy in plants. So it's going to be starch. Number 9, this is an example of a polysaccharide and is used to store extra energy in animals, glycogen. Number 10, the name of bonds between amino acids in a protein. That's going to be a peptide bond. Number 11, ribosomes make proteins based on instructions from DNA. The instructions will go from DNA to mRNA, and then the mRNA will make the protein. Number 12, the instructions for making proteins are located in the nucleus. Number 13, this type of protein helps to move oxygen around the body through the blood, hemoglobin. Number 14, this type of protein speeds up, catalyzes chemical reactions. These are enzymes. Okay, here we have a word bank, and it says match the following organic compounds with their structure or monomer. Write the letter of the monomer and its name next to the name of the organic compound. So a carbohydrate, the monomer is a monosaccharide. And its picture is going to be D. A protein, the monomer is amino acid. And a picture of an amino acid is C. The monomers that make up nucleic acids are nucleotides. And a picture of that is A. Lipids, the monomers that make up lipids are fatty acids and glycerol. And B is a picture of a fatty acid. So again, in number 16, fill in the table below. Be sure to tell me what the monomer, function, and examples are for each biomolecule. So again, for carbohydrates, the monomer is monosaccharide. Again, it's the main source of energy for the cell. It also provides structural support for the cell. Some examples include, we've got glucose, cellulose, glycogen, and starch. For lipids, the monomer that makes up lipids are fatty acids and glycerol. Again, long-term energy storage, insulation. They can also function in cell communication because there are some hormones that are lipids, some steroid hormones, and they also, of course, help make up the cell membrane. Some examples include fats, oils, waxes, like the waxy cuticle on the surface of a leaf. Again, steroids, which are hormones, and phospholipids. The next page. Proteins, the monomers that make up proteins are called amino acids. Proteins have lots of functions. Of course, they help make up your tissues, your hair, muscle, skin, nails, cell transport, cell communication, because some hormones are also proteins. And also some proteins are enzymes which speed up chemical reactions. Some examples include hemoglobin, insulin, and enzymes. Nucleic acids, the monomer that makes up nucleic acids are nucleotides. Of course, nucleic acids, their function carries genetic information that is used to make protein. Two examples of nucleic acids include DNA and RNA. So a little bit on enzymes. Number 17, fill in the table below with words from the word bank to make the sentence make sense. So the shape of an enzyme is very important. If you change the blank or increase the blank too much, the enzyme will blank and will no longer work. So again, the shape of the enzyme is very important. If you change the pH or increase the temperature, too much, the enzyme will denature and will no longer work. Number 18, the monomer or subunit that makes up enzymes are, because enzymes, remember, are proteins, so enzymes are going to be made up of amino acids. Okay, here is a picture of the lock and key model. So which arrow is pointing to the enzyme? The enzyme is going to be B. Number 20, which arrow is pointing to the substrate or the reactants? That's going to be A. So of course the enzyme and the substrate are going to come together, and then the substrate or the reactants are going to form products, which is C. And notice that the enzyme, this is the enzyme before the reaction, and the enzyme after the reaction is unchanged. So number 22, which arrow is pointing to a structure that remains unchanged and is reusable? Remember, reusable enzymes are reusable. That's one of the properties of enzymes. And that's going to be B. In number 23, it says if the reaction in the picture above was hydrogen peroxide being broken down into water and oxygen by catalase or peroxidase, which would be the product? Okay, well, the product is what gets produced. So water and oxygen are the products. Which would be the catalyst? Remember that the catalyst is the enzyme. So the enzyme is going to be catalase. Notice it ends in ACE. Enzymes end in ACE. Number 25, what would be the reactants? The reactant would be hydrogen peroxide. And the chemical formula for hydrogen peroxide is H2O2. And so the reactants, if you remember, are going to be the substrate. So we have question number 26. In the graph below, which line shows a reaction with an enzyme? With an enzyme. So when there's an enzyme present, the activation energy is lower. So that's going to be this first line here, which is going to be, I'll highlight it in with a highlighter. This is going to be the course of the reaction when there's an enzyme present, okay, when it's catalyzed. So it's going to be B, which is the blue line, line B. In the graph below, what happens to the rate of an enzyme reaction as the enzyme concentration increases? So this is just reading a graph. So obviously, as the amount of the enzyme concentration increases, the rate of the reaction also increases. So it increases. Number 28, if an enzyme works best at 30 degrees Celsius, what will most likely happen to the reaction at 70 degrees Celsius? Well, that's way, way hot. And high temperatures will denature an enzyme, will change the shape of an enzyme. So what will most likely happen to the reaction? It will not occur. Because the enzyme denatures, or the shape is changed. So that means that if the shape of the enzyme changes, then the chemical reaction will not take place. Number 29, use the picture below to answer questions 33 and 34. So again, we're looking at a picture over here. Okay, so we got a couple of pictures here. And so we just kind of remember those. It says if the pH of the stomach, enzyme Y works best at pH of what? So enzyme Y works best, it looks like around a pH of 9 or so. Okay, so a pH of 9. And it works best at a temperature. Okay, so what kind of temperature does it work best at? So the temperature looks like around, I don't know, maybe that's about 48 degrees. Okay, and then the next question, the pH of the stomach is close to 1. Which enzyme probably works best in the human stomach? So obviously we're looking something for an enzyme that works well at a very, very strong acid, a pH of 1. So it looks like to me it's going to be enzyme, okay, that's closer to 1. It's going to work, obviously it's going to be enzyme X. So enzyme X would work best. Okay, we got some true false. Enzyme structure is changed during a reaction. That is false. Enzymes are reusable. That is true. They can be reused over and over again. Enzymes work only on specific substrates. That is true. Just like a key will only unlock a specific lock. Enzymes are carbohydrates. That is false. They are proteins, which that leads us to number 35. That is true. Enzymes are proteins. Enzymes work on all substrates. That is false. They are specific. Okay, we've got another word bank here. This one is dealing with photosynthesis and respiration. Fill in the blanks to show what goes in a chloroplast. In other words, what is needed. So what are the ingredients? Okay, what gas do plants